But, okay, that way, anybody can view that later. In fact, that's the other thing. These sessions are on my YouTube channel too, or at least they're in my file, and most of them are on my YouTube channel. So people can always access them later if they can. I can send them the links. Now, Steve, you remember how I know people can actually find my YouTube channel on YouTube if they look. But I tried looking for it, just pretending like I didn't know how to find it. And it wasn't as easy as I thought. So.
Well, I got onto YouTube and searched for Robert Hadaway, and you came right up.
Well, there's also.
It does, but not as Bobby. But it still does even go over to Bobby sometimes.
Well, there's a Robert Hadaway politician in Florida that sort of resembles me, but I don't know if there's any. That's my genes come out of Florida. It could have something to do with that, but half of them do. And then there's I forget his name. He's an actor that did Robert Hadaway in some movies. But anyhow, other than that confusion, that's about it. I think we got all we're going to get because I haven't got any more people trying to get admitted. All
Bobby,
right,
you want me to co-host in case anybody does come in late? I can pop them in.
Yeah, let me do that. Thank you for bringing that up, co-host. All right, let's see. You want to make yes. That way, when I start sharing the screens, actually, I think I actually found sometimes that when I share screen, I can still do it. But anyway, just in case attention gets diverted. Now, sharing screen. I want to try to keep this in a 30-minute range. Let's see. Okay, now still there. Yep. PowerPoint. This, I think, yeah, that's about where I want to start out. Now, this session covers two videos. And there's the last video that's I'm going to make available to everyone if you haven't already seen it. But this, let me get this minimized on my screen. Surely it's not on yours. Okay. The idea here, I was trying to bridge okay, here comes, I actually can admit somebody, Stephen, I just did. So, which is, now this slide is about where I started. Actually, I think it's about 7 minutes into the video, about slide 254 in the total set. But I babble around for about 7 minutes, and then this is where I actually wanted the first video on genetics to start. And the whole idea here has to do, I'm having trouble getting this to, okay, there it goes. This red squiggly line is trying to, all right, this slide bridges the gap between reproduction and genetics. And it goes back and talks about the significance of meiosis, and the first part being just simply the physical reducing the numbers in chromosomes. In the case of humans, from 46 to 23, in the case of garden peas, from 14 to 7. But the most important thing other than that about meiosis is that you get the mixing of chromosomes, and those squiggly lines trying to show you that when the metaphase 1 of meiosis, the homologous pair, here comes Melanie, and the homologous pair line up next to one another in such a way that you get crossing over of the paternal and maternal chromosomes. And at this point, each chromosome is 2 chromatids. And this could be maternal, and the 2 on the other side could be paternal, or vice versa. Now, this is comparing mitosis and meiosis. Notice that in metaphase 1, the chromosomes are lined up next to one another, metaphase 1 meiosis. But in metaphase of mitosis, the homologs aren't paired off opposite one another. And so that's not significant in terms of trying to do any crossing So, but over here, metaphase 1, they're lined up now. And that essentially, they're as close together as they appear here, but you can't see it in this diagram. Now, all right, this is still summarizing, and this is a different way of comparison comparing the two. Reducing chromosome number, the first division separates homologous pair, the second separates chromatids. And you have to understand the difference, what a homologous pair is. Yeah, it's really 4 strands, 2 from the paternal and 2 from the maternal. And ultimately, after meiosis is done, you get separate strands in 4 different entities. Haploid cells with unreplicated chromosomes. At this point, they haven't replicated. All right, now there's the tetrad in metaphase 1 where the crossing over takes place. And now we're ready to deal with genetics. I'm not going to go through the whole thing because that would be repeating 2 videos, but I'm going to kind of run through the slides. This is a pretty good introduction and introductory slide. And I go into the Mendelian genetics first and wind up going into the modern synthesis later. And modern synthesis combines what Mendel learned with what Darwin learned and what we learned in the 1930s with population genetics. And let's see, definitions, basic definitions. And then the intron exon business is not so important. The definition of a gene is pretty good, important thing to try to understand it. It's the piece of DNA necessary to code for a particular kind of protein, or this parenthetical part, more complicated, the other worker molecule like RNA. And the basic building block of a DNA molecule, all right, let's go back. What's the building block of a protein? Amino acid. Well, the building block of a DNA or of a nucleic acid, whether it's DNA or RNA, is a nucleotide. A nucleotide consists of 3 things. A nitrogenous base, like adenine here, thymine, guanine, cytosine, those are the ones. And a 5-carbon sugar, whether it's ribose like RNA or deoxyribose in DNA, and then the H3PO4 phosphate group. Those are the 3 parts of a nucleotide, which is the fundamental building block of a nucleic acid. And these are definitions that you need to understand genetics. Or anybody have questions about those? Mutation is probably one of the hardest ones to grasp because you have micro mutations which operate at the gene level, or you have macro mutations that operate at the chromosome level. And but they fundamentally are both inheritable changes in the DNA. Phenotype is the way something looks. Genotype is the genetic makeup. And if in the case of a homozygous recessive individual, like little r, you can actually, the phenotype will actually show the genotype. Okay, but all right now. This is, okay. What I think, when I see this slide, it makes me think of my line that you are what you are for 2 reasons. Your genetic makeup, your genotype, and the environment you're in. And 2 identical twins can get in different environments and be different phenotypically in the way they behave and in the way they actually look. It's hard to think of behavior as phenotypic, but it's a manifestation of the phenotype. This is a classic. I've got the hydrangea right out in my yard, and you can change the color by the soil pH with the same genes. This is I like too. The aquatic buttercup if the leaves are emergent versus immersed, there'll be difference in the way they look. The environment is modifying the genetic manifestation. That's what's going on in here and in here. All right, let's see what else. Okay, the relationship between it is really simple. Yeah, genotype can refer to a single genetic location or locus, but it usually, when you use the term, it's referring to the total genetic makeup or the genome. Genotype and unique environmental history. Okay, now Mendel was our classic. The thing about Mendel was his research was buried for in 1886 all the way into the early 1900s, and it got uncovered. And so then people started combining it with Darwin's material. They were sort of contemporaries, but it might be a mixed blessing that it stayed buried for a while. The father, okay. Yeah, he was very lucky that he picked garden peas, because if he'd picked something else, things could have been problematic. One advantage is to self-pollinate, and he could cut off the male parts and had control of what was the mating process. He studied 7 different traits, and they were fortunate. They were all independently assorted on separate chromosomes. If they'd have been linked, we might not be having this discussion today. But they were on the 7 different chromosomes, these traits that he studied. Let's see. And flower color is the one we're going to kind of use a lot today, purple versus white. And that's why, okay, purple flowers. The purple flower allele and one white flower, okay, are the 2 variants, DNA variants at the same position on different chromosomes. This is homologous pair of chromosomes with different alleles on the different strands. Actually, they're not double-stranded at this point. They haven't replicated. That's not 2 strands, and that's not 2 strands, not yet. That's before interphase where they get replicated. So a homologous pair could be like this, or it could be 2 strands each after replication. All right. Let's see. Okay, that we'll go into these, or I go into the 2 laws, and I might go to some degree into them today. But let's see. These are the 2 of his biggest fundamental principles: the law of segregation and the law of independent assortment. Let's see. All right, started with a monohybrid cross, where you get the typical 3-to-1 phenotypic and the 1 to 2-to-1 genotypic ratio. The Punnett square, which I go into why I don't like the diagonal approach, but it's still conceptually it's the same. What you have here are your gametes. Your gametes are on top of the Punnett square. In this case, they're showing the male over here and the female here. And when you combine these, you're doing syngamy. You're uniting gametes. Okay. Okay. All right. Why did it take 7 years? Because, okay, he had to make sure he started with true breeding parents, which is part of the problem. And test crossing is one way he went about that process. And the test cross is when you crossed a parent of unknown genotype with one of known genotype, and the only one you really know the genotype of is one that's homozygous recessive. In other words, the parent could be homozygous dominant for purple or heterozygous for purple, but you don't know. It's a monohybrid and you don't know. So, but the way to find out is cross it with a homozygous recessive, yes. And then if they're all turn out purple, you know it was P, big P, little p. If half of them turn out to be white, then you know that the other parent was heterozygous. That's how you know. Okay. I think I'm going to have to get rid of this. Try to get rid of this. This bar gets in the way. If I can move, okay. All right, Mendel's law of segregation. That's each pair of alleles segregates independently of the other, okay? All right, when you go from the parents to here, you've gone through meiosis. That's why here you have a replicated pair of chromosomes, and here you have a replicated pair of chromosomes. But after meiosis, you only have one chromatid. The participant has set speaking language to English. Okay. All right. Maybe I'm speaking in foreign tongue. But anyway, so you have only one member of the homologous pair in the gametes, which is what you would expect. And frankly, in the case of plants, these aren't gametes, they're spores, but we won't. And then they unite, and the gametes have to be the one to unite. So technically, the bottom line is the spore is somewhere in between this, and it's not showing. But in animals, there's nothing in between. It's just here to here. But when gametes unite, and that's how you know it's not a spore, because spores don't unite, they divide. You restore the diploid condition, and so we wind up with a dihybrid F1. And then the F1 gametes, when they cross, you go into an F2 and you get into the Punnett square. Let's see. Now, the second fundamental law, there's more than two, but the second was the law of independent assortment. And what that has to do with that the chromosomes, the traits sort on different, in different gametes. And let's see. All right. And what this has to do with is it's not independent assortment. And the reason it's not independent assortment, and this is a slide that got added into the set. If they're linked, the traits can assort independently. Like in this case, they can assort independently because they're on separate chromosomes. So that's really what independent assortment means. If they're linked on the same, and by the way, this slide got added separately in the set and wasn't in the original set. This was, but these two are two different ways of looking at, actually the one on the right is the same as this, but in that instance, the traits of color and shape can't sort independently if they're linked on the same chromosome. The only way that can happen is if the traits sort independently because they're on separate homologous pairs, which is why Wendell's was lucky and got his all 7 traits were on separate pairs of chromosomes. Okay, let's see. This slide has some add-on to it, and this is an important rule that 2 to the nth power will n equal the number of heterozygous pairs of chromosomes. That's how, if there's 2 heterozygous traits, that means 2 squared is 4. That means there's 4 different kind of gametes, and that's why you got 4 here and you got 4 here, and then when they start doing syngamy, you wind up with the dihybrid 9:3 to 3:1 ratio and the genotypic ratio of 1:2 to 2:4 to 1:2 to 1:2 to 1. And if you could go through this, you could count those numbers up. And what this is trying to do is point out that there's a little glitch in the nomenclature of the phenotype. Smooth and round are the same thing. And so between these diagrams are the same. It's just that the nomenclature is round instead of smooth. Actually, it says round here, and then here it says maybe smooth, but the bottom line is smooth and round are the same thing. Let's see. Let me get this thing moved again. All right, yeah, these, besides the independent assortment and segregation, he came up with other concepts, including law of dominance, where one trait can totally mask the other. He didn't know about, unfortunately, didn't run into incomplete dominance, which is something that's going to be talked about in a minute. He was lucky about the traits were unlinked. He was lucky that he had complete dominance, and that each character is controlled by a single gene, which is not necessarily the case. Incomplete dominance, I think, is the very first next slide. Polygenic inheritance is after that. That's like height. So all of these, except for epistasis, which is, I don't go into further discussion much, except for what you see here. One pair of a gene pair controls gene pairs at other loci, other points on the chromosome. It's kind of like epi means on top of, hypo means beneath. And so a pair of genes actually controls another pair of genes. And the one that gets controlled is hypostatic to the epistatic. So luckily, Mendel didn't have to, if he'd have run into these complications, he'd have probably gave up. Now, this is the classic example of incomplete dominance. In snapdragons, you have 3 phenotypes that match up with the 3 genotypes. Red, pink, and white. In the middle are heterozygous. So that's the case of where the phenotypes and genotypes do agree. What you see is what you get. What you have genotypically is what you get phenotypically. And height is polygenic. Lots of genes control height. It's the classic example in humans of polygenic inheritance. And in tobacco, height is similar. This talks about pleiotropy, one gene affecting multiple traits. He's lucky about that. Let's see. All right, and then the modern synthesis is when you combine what Mendel did with what we learned in the 1930s about population genetics, and then it got put together in the modern synthesis. All right, let's see. All right. Anybody got questions? Have I confused anybody? Or did you have questions when we started, which is what I should have started by asking? All right. Plant asexual reproduction is we talked about it being a short-term advantage but a long-term liability because you don't get variation. But the sexual reproduction is our focus when we talk genetics. I define propagule early on, but don't talk about propagule dispersal till the last, pretty much to the end. I think it's not even till the evolution part. All right, propagule is any plant part, asexual or sexual, that becomes detached from the parent plant that can form a new plant. And technically, in the presentation, I talk about the fact that we're talking about myospore, M-E-I-O-S-P-R-E, not a mitospore, because a mitospore would be the product of asexual reproduction. And in bryophytes, there's some of them have structures called GEMMA-E, plural of gemma, that are asexual propagules. Or some of them have miniature plantlets called brood bodies, but those are both asexual propagules. Now, pollination, you can't have fertilization without pollination. And so that's why this is where it is. Getting pollination is about getting the pollen from one sex to the other sex, whether they're on the same plant or not. All right, and then I talk in the presentation about the majority of angiosperms are pollinated by biotic mechanisms, and today the minority is by wind and water, like 2 or 3% water and and around the 8 to 10 are wind pollinated. And the wind pollination in angiosperms is believed to be evolutionarily advanced. In other words, oaks and willows are wind pollinated, and that's believed to be a return to the way their ancestors, like, for example, gymnosperms are primarily wind pollinated. And I talk about the fact that in the tropics, it doesn't pay to be wind pollinated. You're not, because your pollen is hardly going to get to from point A to point B because there's foliage blocking it. And so the further you go toward the equator, toward the poles, the more wind pollinated you're going to be. And so I even got a slide somewhere here. Yeah, here. You can look in the background in February in Pennsylvania, and there's not going to be foliage blocking. This poplars and willows are the same family, they're dioecious. And so getting the pollen from one plant to this female plant, which are made up of pistillate catkins of about 50 flowers in each catkin, you have to, it pays to do it in the wintertime or early in the spring, before the leaves are blocking the passage. And frankly, let's face it, there's not going to be too many insects running around in February in Pennsylvania. This and the next, those slides are about the 2% of angiosperms that are water pollinated, which is very rare. And by the way, that bullet, I've already explained that some angiosperms like oaks and willows are secondarily returned to wind pollinated. Primitive angiosperms are animal or biotically pollinated. Now, I did mention that willows have hedged their bets by having a gland which could attract biotic pollinators if wind fails them, which is pretty sophisticated to be able to do both. This is pretty pathetic for a flower, isn't it? Down to a bract. There's not much showiness to it, but you don't really try to track too much. You're interested in wind pollination. Oaks, classic example of monoecious, where you have the male and females on different branches. Most people never see these little female flowers. They're just down to that stigma, style, and ovary, and there's no petals or sepals. Really, so very basic. If you ever get a chance to go to the Chicago Field Museum of Natural History of Chicago, you should, because there's a male model, there's a female model. They're glass. Hard to believe they're glass models. I used to have my students go to their part of their labs where to go to that when I taught in the Midwest, and they spend 2 days there, 4 hours each. Okay, the monocots like graminoids like grasses and sedges and rushes are primarily wind pollinated plants. And these are comparing the two. Obviously, wind pollination is going to have a lot of wasted pollen. Like pretty soon, I'm going to have to power wash my garage out there, my porch outside, because the wind is going to get pollen all over everything from my oaks. And I have a pine plant, not mine, but across the road, I have a pine plantation that's going to contribute pollen. It's not already. And then in the backyard, I've got a bunch of volunteer pines that are just going to do the same thing. Dump massive amounts of pollen. All right, insect pollinators. Let's see. There's nothing really heavy about these. Are just different examples of how biotic pollination goes on. Now, interestingly enough, I heard a NPR thing the other day talking about the fact, of course, humans can't see ultraviolet in insects, and lots of other animals can see ultraviolet. I'm surprised at how many animals, other than even insects, can see ultraviolet. But that's where your nectar guides come in. Um, All right, this I talk about in the presentation. I've got a plant. This is in Florida, moonflower, pollinated at night by a moth with a very long tongue. But I've got in my yard another Ipomoea macrorrhiza that's the original came about 4 miles from here on Fort Stewart, and it's also night pollinated. This, the largest flower in the world, is a parasite, a root parasite. And then this is a succulent that's in the milkweed family or the dogbane family if you lump it. But the point is they both have that fleshy, rotting flesh color, which has to do with the fact that they attract flies. And I've smelled the one on the right. I've never been to, even in a conservatory, seen that one, or at least not smell it. I think I've seen it. But, all right, birds like color, and this is no surprise here. That's pretty much, we're about out of time here on 30 minutes too. Let's see, breeding, let me move this. Yeah, breeding systems. I get into, I'm starting to get into the last lecture on evolution. Okay. Inbreeding, some flowers are cleistogamous or closed and they self-pollinate. So this goes to the adage that any sex is better than no sex, but ultimately it's not good for long-term advantage. Chasmogamous is the norm. Cleistogamous is self-pollinating closed. And these are having separate representatives facilitates outcrossing. Maturation times is a way to favor outcrossing, and like pollen getting ready on one plant before the female's on the same plant. And this thing is flowering right now. It's a state flower of South Carolina, and the common Gelsemium or Carolina jasmine. Some plants, I never can remember which is strum and which is pin, but I just think of long style and short style, and that's a mechanism that favors minimizes self-pollination. And if you look around, you'll find both in a population. I don't remember which is predominant. I've got it in my yard. This plant just is well, vines, any vine in your yard is going to ultimately be a lot of maintenance, I'm telling you. All right, so this is more into breeding systems. Okay, let's see. After selection, okay, now we're getting into evolution. Let's see where I'm at. Well, let me try to back out of this slide. Oh, okay, finally. All right, slide 348. Yeah, I've already gone into pretty much a lot of what I'm going to go into the next time. But slide 323 is about where this second video actually stops. So I started getting into I thought I ran a little over. But, all right, yeah. I define propagules here, but I don't talk about propagules until the last session, till the very end. So I got in ahead of myself a little bit there. But anyway, I've gone a little over 30 minutes. I hope I haven't wasted your time. So anyway, all right, are there any questions?
I popped one into the chat box earlier, Bobby. It's kind of a vaguely related point of curiosity.
Let me see it stop the sharing. I think I have to do that to chat. Okay. Steve, let's see. All right, oh, wait a minute, okay. Is that it? Thank you. I'm looking for it, Steve, I don't see it. Oh. Huh. All right, Steve. Let's see, type message here. Is a term, oh, here it is. There's a term allele in any way related to, let's see. What? I don't see what the rest of it is. Steve, you want to repeat it?
Sure. I was just curious about the term allele. Is it any way related to the term alleliopathy?
Yeah, alleliopathy is you said it the Latin way, alleliopathy. But that's the only thing I can see is the term allele related anyway too, and then I can't see the rest for some reason. But allelopathy is a phenomenon that's basically talking about plants producing having the genes that enable them to produce toxins against competitors, and sometimes their own offspring. But usually it has to do with plants producing allelopathic chemicals that suppress the growth of other plants and minimize competition. And that's the concept. So, all right, I'm surprised for whatever reason, I don't understand why I couldn't see that whole question, but thank you. One new message here. Thank you, Bobby. Okay. All right. All right. Well, I'm not operating on all cylinders today for some reason, but I don't know why, but I guess I'm just, my birthday is coming up. I'm soon be 75. That's my excuse next week. But anyhow, all right, as soon as I'm done, I'm going to go send everybody, well, actually, I'm going to save this and it'll take time to process, and I'll put it up as a YouTube channel for one or two people that I know are going to want to see it. It couldn't be here because of the time I chose. And the other thing is before the day's out, certainly before tomorrow, I'll make the last session available. So we'll have one more question and answer session, and then we'll be done. And this thing went from January to August of 21 when I first did it. But anyhow, let's see, the last session doesn't really start till about, so when I put up that last evolution discussion, it doesn't really start till 4 minutes into it if you want to cut to the chase. I looked at that. All right. Bye, folks.
Another chat from Melanie just popped in.
Okay. See that. All right. So Melanie says in the term, Steve. For some reason, all right, I finally see the rest of Steve's. Now, I'm just wondering what's wrong with this. Every time I think I've got, you know what? Every time I think I got this figured out, something happens. I can't see Melanie's chat. Okay, here it is. I tend to take, okay. Yeah, Melanie says she intends to take the entire course for retention. Will you be offering the course again? Now, I answered your email, Melanie, and saying the short answer is no. I didn't say that in the email. I won't be offering again. And I don't have to, because the main thing is, if you look at the email I responded to you on about just before this class, I give you what you need to do. And it's basically is access to materials that are in the links I provided. And I gave you the link to my YouTube channel. So basically, you can access all of these sessions, question and answer. You can access all of the videos. You can access all of the slides. The only thing you may have to, if you didn't save them, you may not have the handouts that aren't slides, and I can send them to you as you need them. And that would be when you come across them in the slides presentation or the video that says handout on, for example, I gave matching exercises, and those are non-slide handouts. But basically, I won't be re-offering the course again. All right, that's because you can self-pace it the way I've done it. All right, anything else? All right, bye, folks.
All right. Bye.